Good afternoon. Happy solar eclipse day. My name is Alfredo Fontanini, and I'm the chairperson of the Department of Neurobiology and Behavior. So thank you for joining today's lecture, whether you chose to join it in person or to join our live stream. And welcome to the 26th Schwartz Foundation Mind-Brain Lecture. So this lecture is hosted by the Department of Neurobiology and Behavior and is generously funded by the Schwartz Foundation. So let me tell you a few words about the Schwartz Foundation. So it was established in '94, 30 years ago, to support and promote theoretical and computational neuroscience. I'm sure most of you know what that field is, but in case you don't, this is a cross-disciplinary field that really brings together physicists, mathematicians, engineers, computer scientists, and neuroscientists. And the goal of computational and theoretical neuroscience is really to understand the relationship between the brain circuits, and the mind. Now, the Schwartz Foundation was really a pioneer in supporting the birth and the establishment of this field, and was really instrumental in mediating its success. The Schwartz Foundation is the brainchild of Dr. Jerome Schwartz, Jerry. Jerry is a physicist and an engineer. He is the co-founder of Symbol Technologies, and is an emeritus trustee of the Stony Brook Foundation. The Schwartz Mind-Brain Lecture, this lecture, was created in 1997, and the idea was to have a lecture that would advocate for a computational perspective on the brain and introduce fundamental challenges in the field of neuroscience to our community, to the campus and off-campus community. And throughout the years, it brought to neuroscience exceptional scientists, presenting their research into the most significant fields of neuroscience, and particularly the fields that impact everyday life. This year's distinguished speaker is Dr. Sabine Kastner. So Dr. Kastner received her BA in History and Philosophy from the incredibly prestigious and historical University of Göttingen in Germany, her MD from the University of Düsseldorf, and her PhD in neurophysiology from the University of Göttingen and the Max Planck Institute. So she joined the faculty of Princeton University in 2000. She is currently professor of psychology and neuroscience at Princeton. She's also the scientific director of the Scully Center for the Neuroscience of Mind and Behavior, and she's the director of the Silvio Conte Center for Basic Research on the neural mechanisms of decision-making and attention. Dr. Kastner is a very accomplished neuroscientist whose scientific work unveiled the brain networks and mechanisms responsible for attention and visual attention. More broadly, her work has established fundamental principles linking brain structure and function to cognition. She has published more than 150 articles on incredibly prestigious journals, and she has been cited almost 30,000 times. And so anyone who's in the business of publishing for neuroscience knows what a large scale of citations that is. Dr. Kastner is the recipient of numerous awards, including the George Miller Prize in Cognitive Neuroscience, the Society for Neuroscience Award in Education and Training, and she's a fellow of the American Academy of Arts and Sciences, the Society of Experimental Psychology, and the American Psychological Society. She's also a member of the German National Academy of Sciences, Leopoldina. This is a society that was founded in the 1600s. Finally, and this is, I think, is a very, very important point, it's worth mentioning that in addition to her monumental scientific contribution, Dr. Kastner is well known in the field for her service to the community. She's extremely committed to outreach through teacher seminars, public school lectures, and engagement with support groups for parents of patients suffering with neurodevelopmental disabilities. Last but not least, she is the editor-in-chief of the Journal of Neuroscience, one of the most prestigious journals in the field and the official journal of the Society for Neuroscience. And she is the co-founder of Frontiers for Young Minds, a journal to outreach and to promote neuroscience knowledge for middle school and elementary, middle, and junior high school students around the world. Today, Dr. Kastner will tell us about her groundbreaking work on the neural mechanisms of attention. And so please all join me in welcoming this year Schwartz Mind Brain speaker, Dr. Sabine Kastner. Thank you.
Good afternoon. Thank you very much, Alfredo, for this very kind introduction. This is certainly a special lecture, not only because it's a brain-mind lecture, but because you arranged it in a way that it coincided with a solar eclipse. So I will certainly remember that for the rest of my life, and I want to thank all of you to come to this lecture instead of watching the resolving solar eclipse. So I'm really happy to have this opportunity to share the research that I have pursued for the last 25 years or so with you today, at least a few pieces of it. Now, I want to start with a very famous citation by William James from his book, Principles of Psychology, that was published in 1890. James was the father of psychology in the US, and his book has an incredible insight that is still feels very modern and interesting. So this is what he says about attention. Everyone knows what attention is. It is the taking possession of the mind in clear and vivid form of one out of what seems several simultaneously possible objects or trains of thought. Focalization, concentration of consciousness are of its essence. It implies withdrawal from some things in order to deal effectively with others, and is a condition which has a real opposite in the confused, dazed, scatterbrain state. So there's a lot of information about what attention is in this citation, but I want to just allude to a few things that I find particularly important that he notes here. So he says that when we select something using our attention system, we can select one thing at a time of several things that may be available to us either in front of us, these are the objects, but he also talks about internal representations, the trains of thoughts. But we have to select one and basically leave everything else behind. So this is the withdrawal from other things that he also alludes to. And this is what he gets here. He doesn't use these words, but what he really gets at here is, I think, what is at the core of what attention function is and why we have it and why we need it. And that is the incredibly limited processing capacity of our brain. This is why we have to select one thing and withdraw from others. So I want to illustrate that to you. So here's a scene, a sailboat scene. It has a lot of things going on there, sailboats, people, and so on. And you study this scene now, and if I would withdraw it right now and ask you questions about it, you may think, yeah, I know every single detail in the scene. It's not so complex after all. So I don't believe this entire thing about the limited processing capacity. So let me see if I can convince you here. So I have a demonstration here. There will be a movie. It will go back between two frames of the sailboat scene here. And there's a change in between. It is not a small change, just to give you that hint. And I will ask you to detect the change and show me your hand. Please don't give it away, okay? Don't spoil it. Just show me your hand once you have detected the change. So, I'm also timing this, so I want to see, okay, I see one hand thus far, two. We are about almost 10 seconds into this demonstration, and most of you have not discovered the change. A few more. I think we're nearing halfway of the audience, so here's what I'll do. I will draw your attention to the horizon. Look at the horizon. Did everyone get the change? So once I directed your attention to the horizon, you were able to overcome this limited processing capacity. If you were able to represent this information all at once, you would have detected that change right away, but you didn't. Once I drew your attention to this location here in the scene, you were able to overcome that right away. And that is the power of this attention system that makes these selections for us. Now, I show you a second one, just to make the point that you cannot learn these kinds of tasks very easily. So it's the same thing. There's a big change here in this scene. Show me your hand once you have it. So a few of you get this. This went a little faster. The change is also much more salient. Who didn't get the change yet? Okay. Look at this. This person is changing his pants in front of you constantly, but you missed it, even though this was there for quite some time. So there's limited processing capacity in the visual system, and this is why we need these mechanisms for selection of relevant information that I'm going to talk about today. So let me just build on these ideas a little bit more with this piece of art by one of my favorite visual artists, René Magritte. So when we select information from a scene like that, what we can do is to recruit a spatial selection mechanism. In our textbook, we talk about this as a focus of attention or a window of attention or a spotlight of attention. It has a lot of metaphors. But what the spotlight is doing is you can basically allocate it somewhere over a limited amount of visual space in front of you. And then you take in all of the information that falls into this spotlight in a preferential way. What you can do with the spotlight is you can move it around. You can move your eyes with it. That is what we typically do when we search through scenes. That would be an overt shift of attention. A shift of attention with moving the eyes. We can also do that covertly by fixing our gaze here, let's say, and then covertly direct our focus of attention to that location. So we dissociate our gaze from our focus of attention. We do that all the time when we drive, for instance. We are looking straight, but we are monitoring the periphery of the visual field so that we don't miss if anything happens there. We would call this a covert shift of attention, so a shift of attention without moving the eyes. And I will tell you today in my talk a lot about these covert mechanisms of shifting attention around. Now, my story today will have 4 chapters. I will tell you a little bit about the behavioral measures of attention in the laboratory, where we typically, unfortunately, do not study Magritte's artwork as attention researchers. I will then tell you about the brain network for attention function that subserves these kinds of selections, how this brain network is coordinated. And then I want to tell you a little bit about the future and what's going on right now, not only in the field of the neuroscience of attention, but more broadly, I think, in the field of cognitive neuroscience. And I think it will lead to a revolution of how we will view brain activity in the future. And those are the dynamics of cognition. Okay, so let me start how we study this. In my laboratory, we study 2 primate brain models, the human brain that you see here on the left and the macaque monkey brain on the right. And we use behavior to glue these 2 brains together. So typically, we will use a very established task from human cognitive psychology, and we will try to train macaque monkeys in this task. And if we are successful, we can then use that behavior that is similar to glue these 2 brains together and to relate the neural signals that we can measure in different modalities with functional brain imaging methods or with electrophysiology. I will give you examples later on. And we measure them in these two brains at different levels of analyses. And the behavior can glue these signals then also better together in meaningful ways. Now let's start with the behavior that, or the behavioral measures of attention that we typically study in the laboratory. So here's a typical attention task. We ask our subjects to fixate centrally here on the square. In this particular task, we show two bar-shaped objects. They can be oriented vertically, like in this example, or horizontally. This will change from trial to trial. Then we draw attention to one of the four corners of these objects. So here it would be the upper right, but it could be any of the other corners. Again, that will change from trial to trial. And this cue will be presented very briefly, and it really is very effective in drawing attention while keeping the gaze, the eyes over here, to this location and space, and to maintain attention there. Then there's a brief delay, and at an unpredictable time, there will be a target, a very dim one, much dimmer than this one, at threshold, at perceptual threshold that we show here in the cued location with a high probability, let's say with 80% probability. And sometimes in these 20% of cases, the target will be, at the other location here, at the opposite location from the cued location. So we call the cued location the attended location, and then compare with the behavior at the non-cued location. We call this here the unattended condition. Now, what we typically see is, first of all, that you can react faster to the target in the attended location and the cued location. So let's say you can make a decision and reach a motor response within 300 milliseconds. At the unattended location, that would be on the order of 350 milliseconds. So it's not a whole lot, but it's something that you can definitely reliably measure. You can also show that the accuracy at the cued and attended location is much greater. So here, let's say it would be 85% of the time, and at the unattended location, it would be about 70% of the time. So these are the typical measures of attention that we have in the laboratory and that we can work with. Now let me summarize what we have thus far. So the brain's capacity to process multiple items at the same time is severely limited. Attention mechanisms serve to counteract limited capacity and process selected information preferentially. And at the behavioral level, this is reflected in faster reaction times too, and greater accuracy of attended information. Now let's look at the brain. So I want to introduce you to the brain network that subserves these kind of spatial selections that I've shown you thus far. So here's an animation. This is a movie, and I will tell you what you see in a moment. We are looking here at a right hemisphere, right half of a brain. It's a human brain. And this is a cortical sheet, and we basically inflated it. So the real human brain has a lot of windings, but then it's very hard to appreciate what goes on inside of these windings. So you can think about this inflation process as if you inflate a flat tire, basically. So this is the posterior pole of the brain. This is where the visual system or many visual areas reside. And this is the frontal pole, and we're looking here kind of from a bird eye perspective on this right hemisphere. So I'll show you functional magnetic resonance imaging signals that float to this brain during an attention task. It's really very simple. It's just the allocation of attention in space, making a detection similar to the behavioral task that I have just introduced to you. So this is a visual task. What we see in the beginning, everything that's red, orange here, are fMRI signals. So they float to this brain, initially through the visual system here in occipital cortex. And then later on, there is a buildup of activity around this area here. This is called the parietal cortex. And then also in frontal regions that are more difficult to appreciate in this particular view. So there's a huge brain network engaged doing something simple like this allocation of attention in space. It encompasses regions in all major lobes, a lot of subcortical regions that you cannot even appreciate in this view. And the fundamental problem that interests us is how this network produces a behavioral response within 250 to 300 milliseconds. It seems like a huge deal given how much is involved here in terms of real estate of the brain. So these kinds of networks are not unique to attention. You can show them for any cognitive function that you want to study. So all cognition in the brain is organized into networks, and we need to understand how these networks operate if we want to understand cognition. Now, let me start with an old experiment that I did when I was a postdoc in Leslie Ungerleider's lab at the NIMH in Bethesda. Leslie and I did a very simple experiment. What we wanted to understand was whether there was functional organization in this enormous network, attention network, that we observed in our studies. And we did a very, very simple experiment. We directed attention, this focus of attention is just a schematic here to illustrate where attention was allocated. That was marked here with an arrow at fixation. So this is where the eyes are in this experiment. So we directed attention to this location in space and had the subjects expect the onset of the stimulus. And it was unpredictable in time, and it was a long time, this expectation period. And then we showed them an area of stimuli of these 4 stimuli, and our subjects would detect one of the 4 stimuli and react to it in this particular location closest to the fixation point here in the array. And what we saw was quite interesting. So this is an fMRI experiment in humans, and what we saw in visual cortex, so this is visual area V4, so it's like a mid-level area in the visual system, but we saw that also in many other visual areas, was that during that expectation period, when we just have the allocation of attention in space, there's a large increase of activity. That's this yellow period here. So there is no sensory, no visual drive to this system. It's just a cognitive drive, a cognitive signal that gets represented here. And then once the visual information kicks in, there's a very large increase in addition to that. Now, what we saw in other areas, in what we call higher order cortex, in frontal and in parietal cortex, I'll give you an example here for the human frontal eye field, was very different. So here we saw a larger increase during this expectation period when attention is allocated in space, but then that was not really modulated when the sensory information kicked in. So that was sustained as long as attention was directed to this location and then got back to baseline once attention was redirected to the fixation point. So this activity in frontal and also parietal cortex seemed to reflect the cognitive operations that are involved in this particular task and not so much sensory processing. So we saw these 2 different patterns. And from experiments like this and many other experiments that people have done in the field, we came up with a very simple organizational schema for this attention network, where we basically distinguish what we call sensory, or here it's visual modules, because we're in the visual modality in these visual regions here, and they're different from cognitive modules that we see in frontal and parietal cortex in this example. Now, what functional magnetic resonance imaging and other imaging modalities are really wonderful for is to illustrate these networks and to kind of make gross organizational distinctions like the one I've shown you. But what you cannot appreciate with these methods are interactions between these modules. You cannot infer how these regions all interact with one another because the temporal resolution of these methods is very low. So for that, we turn to electrophysiology. And we record in my lab sometimes directly from the human brain. And I want to introduce that to you. These are from typically epilepsy patients who get implanted with electrode arrays. You have two examples here from intraoperative settings. And this is done to prepare these patients for their neurosurgery to remove the epileptic tissue. So this is required for diagnostic procedures and does not have any scientific purpose. So we as a scientist are just the very humble bystanders, and we get sometimes lucky when the patients are well enough and willing to participate in our experiments to get recordings directly from the human brain, which is a really, really amazing experience for neuroscientists. So we are very, very grateful to our patients to have this unique opportunity. Now, when we study attention function with intracranial electrophysiology, we take advantage of a fundamental principle that we have in the visual system, and that is that there is spatial representation. So visual space is systematically represented there. I want to illustrate that to you. It was a very simple experiment. So here, our patients will just keep their eyes fixed here on this cross while we are showing this light disk in these locations around the visual field, so in different locations around the circle, basically. Now, while we are measuring activity in their brain, so here the disk is shown at this location, and all what you see here in these bright colors would be the equivalent of electrical discharges of hundreds, probably thousands of neurons that are underlying this particular recording site. So you can think about this as multi-unit activity. Now here you see at this location, there is a very strong response. And now when you go to neighboring locations, you will still get pretty strong responses, not as strong, but it's pretty good. And when you go further away, there will be less and less. And then in these locations here, there will be no responses. So from a simple experiment like that, you can reconstruct what we call a response field. You may have heard about a receptive field. So this would be the equivalent in the human brain. And this response field represents this particular space and nothing else. And so in an attention experiment, we can take advantage of that. We can show some visual information here, some kind of stimulus, while we draw attention to that response field, or we draw it away, and we can now look at how this information that we're showing there is processed under conditions of attention to the information when it's processed in that response field or away from it. And when we do a simple experiment like that, we can learn a whole great deal about the interactions of sensory and cognitive modules. So here we have a recording in area V2. So that's an early visual cortex, the second visual area that we have in the system, in the human brain. And we are looking here at something very simple. It's just the processing of the shape stimulus here. This is the response field. And the red line is the condition when attention is drawn to that response field. And the blue one is when attention is drawn here, but the shape is still there. So this shape is just unattended. Now let's see what we get here. So in the beginning, you see that during this period over here, the two responses are overlying on each other. So there's no difference whatsoever whether a stimulus is attended or unattended. But then around 270 milliseconds, these two lines deviate such that the attended information gets a boost. So it gets a response modulation, and the modulation is an enhancement of the response. But this occurs with a significant delay. And this is the most classical response modulation that we find in the nervous system by attention, this response enhancement, this boosting of a response, which probably reflects this preferential processing that I talked about a little earlier. Now we can oppose that to looking what's going on in a cognitive module. This is in parietal cortex. So here you can see that this response modulation is much, much stronger. So this difference here is much greater than this one over here. And you can also see that it starts much earlier, this deviation of the blue and the red lines, the attended and the unattended condition. So here basically attention already does something to these neuropopulations at about 140 milliseconds. Now, from an experiment like that, just looking at these latencies, we can basically infer that the cognitive module here may actually send some kind of feedback signals into the sensory modules, which could then explain this delayed occurrence here of this response modulation in visual cortex and the sensory module as opposed to the cognitive module. So from experiments like that and similar studies, what people have proposed in the field is what we call a top-down model of spatial attention. So the idea is that we initially have this flow of information here to our shape stimulus to the entire system in this bottom-up driven way. And then the cognitive modules here are feeding signals back into the sensory representations and basically modulate the ongoing processing in the visual cortex. And this is a pretty standard model that you will find also for a lot of other cognitive functions, so it's not unique to attention function at all. But this is kind of how we think about the relationship of the sensory and the cognitive modules. Now, let me briefly summarize this second chapter. So I've shown you that attention function is subserved by a specific, dedicated, large-scale network. And this network is organized into frontoparietal cognitive modules and occipitotemporal sensory modules. Frontoparietal regions control processing in visual regions via top-down feedback signals. So the next chapter that I would like to discuss, or the next question, is a very, very fundamental question for the entire field, and it's a very unsettled question. So I will just give you kind of my answer to what I think is a really fundamental question. And that is, how are these networks coordinated? I have shown you that they consist of an incredible amount of different areas that are engaged. Attention function is very, very efficient. It is fast. How can information get routed through this large-scale network in such a short period of time to give rise to behavior? So I want to kind of simplify the problem by just thinking about how two areas within that network may get coordinated. So I just call them here area A and area B, and they are, happen to be in the cognitive modules here. So let's think about the problem. We have a spike train, so a train of spiking activity, electrical signals that were produced here in area A. How can that information get to area B? So one thing that we know for quite some time is that spiking activity has temporal structure. So this, is not a coincidence here that it's shaped like that, but it has a temporal structure, and the temporal structure can be revealed if you look at oscillatory activity, that is population activity that you can measure in field potentials. It may look like something like this oscillatory activity. And here the spiking activity coincides with the peak of this oscillation. And so this oscillatory activity in these regions basically sets windows of greater excitability during which spiking activity can be produced, and lower excitability during which it is much less likely for a spike to emerge. Now, if the spikes here want to travel from A to B, it is advantageous for them to align, so the traveling goes in this direction, to have oscillatory activity that, again, can parse out these excitable windows for area B with an appropriate phase delay to synchronize with area A, so that something like that would emerge here. So a lot of the spiking activity could get transferred that way when this oscillatory activity here gets synchronized. If it's not synchronized, we would have kind of a situation like this, and you can see that very little of this information can get to area B. So this is, a very simple way of thinking about the problem of how two areas can communicate. Now, the question then is, how can they synchronize activity across these two populations? And one way to do that is to use the cortical-cortical feed-forward and feedback pathways that they share. So this is just the assumption here that these two areas share these feed-forward and feedback pathways. So they could basically do like a local negotiation, what a good state of communication would be for them, or a state of neural synchronization in this model that I'm using here. Well, if you think about that at a network level, that may not be a very elegant computational solution to the problem, because it would probably take a lot of time to have all these local areas kind of negotiate something that also then materializes in a coordinated network. An alternative solution might be if a third area that is interconnected with A and B might do that synchronization for them. This would be this area over here. It's actually in a region that's buried underneath the cortex and the center of the brain. So this is like a look-through. It's called the thalamus. And thalamic regions are interconnected widely with cortex. And I will show you in a moment that they could play such a role. If they are also interconnected with other parts of the network, they could really do this coordination for the entire network in basically a very efficient way. So that's the idea here. And so we have studied whether the primate thalamus can play such a role, particularly higher. We call them higher order nuclei because they are interconnected with the cortex and not with the sensory periphery. And we have studied one such nucleus extensively. And that is the pulvinar nucleus of the thalamus. That is this large nucleus here that's in the posterior thalamus. The pulvinar is the largest nucleus in the primate thalamus, and it expands during evolution on the same order as prefrontal cortex. It is widely interconnected with the cortex, and the connectivity is actually quite intricate. I will not give you all the details of this schematic, but the punchline is that any two areas in cortex that have a direct connection, that would be this line here in the schematic, like these two areas that I highlight here. Any two areas that have a direct connection also have a transthalamic loop that goes through the pulvinar. So that would be this pathway over here. So think about the heavily interconnected areas that we have in these networks. They all have these loops through the thalamus, and we wanted through the pulvinar. And we wanted to understand what the function of these loops is. And what we found is that they communicate in a certain frequency band, and that's a 10 Hertz frequency band. So all of these interactions here, the transthalamic ones, as well as these ones during an attention task, are typically organized around a 10 Hertz oscillation that you can measure, again, in a population activity like a field potential. So it looks something like that. This is not from my experiments. This is from the first recordings of 10 hertz activity, alpha activity in the nomenclature of electroencephalography. This is from the first recording that was ever done, and where the alpha activity was described here by Hans Berger in 1929. But this is basically how our activity looks like. So you measure in the population activity these regular cycles that go at about 100 milliseconds. So this is the communication frequency that this network is using, and you can see it here. You can see that attention influences these. So here we're looking at the synchronization between the pulvinar and a cortical area. So that would be, one part of this loop here. When attention is allocated at a response field or at the opposite side, and you can see that we see this very nice strengthening of the synchrony between these two areas in that alpha frequency band. And we can also show in these experiments that the pulvinar is actually synchronizing the cortical areas in that frequency band, and again, that is modulated by attention. That's what you see here, whereas the feedback and feed-forward pathways that these two areas share do not get engaged. So this kind of synchronization is really something that the pulvinar imposes on the cortical network. And we think that this is a very elegant solution to the problem of network communication. So we think about the thalamus, again, the pulvinar is doing that for many, many parts of this network. And we have shown that in many, many parts of this network, this synchronization. So we think about the structure that's an old structure deep in the thalamus as a sort of a timekeeper. It organizes the cortical networks in time so that they can efficiently process information. So what I showed you in this part of my talk is that the pulvinar regulates synchrony between cortical areas during spatial attention, and we think that the pulvinar may control the temporal dynamics of cortical large-scale networks in form of a timekeeper. And we think that the pulvinar is not only doing that for attention function, but probably for many other cognitive functions as well. It is still a very understudied area of the brain. So if you want to make discoveries, go to the pulvinar. It's a good place to make discoveries. Okay, so in the last chapter, I want to introduce you a little bit to where the field of the neuroscience of visual attention is at this moment in time and where it's going. So I think this is kind of the future that just has started, and we just learned like the tip of the iceberg of something very, very exciting. And these are attention rhythms. Now, when I introduced the spotlight of attention to you in the beginning, I had told you that it has a certain spatial extent, and we can move it around. But what we perceive subjectively when we allocate attention, so you may allocate attention right now on my slides or on your phone or on me, but wherever your attention is, what your subjective experience is that you take in information continuously over time. And I will show you that that's just an illusion. What really happens is that our attention focus is blinking. It goes to cycles where we take in more information and cycles where we take in less information. And so I want to tell you about these attention rhythms that we can measure behaviorally as well as at the neural level. So here's our attention experiment. I introduced you to this experiment already at the beginning of my talk. So just a recap, we have a fixation point, these two bar-shaped objects. We cue here in this trial in the right upper corner, then there is a delay during which attention is allocated at that cued location. And then there will be the detection of a target at threshold, a very dim one. It's a very hard task. And so we wanted to understand what's going on during this period of time here. So we varied what we call the cued target interval systematically from 300 milliseconds to 1600 milliseconds. So I plot now the behavioral outcomes, or the ability to detect the target as a function of that cued target interval. And this is in humans. Now, what you can see, so this is the change in hit rate. This is the cued target interval. So if you were to do this task right now, I would be able to tell you exactly when you're good at the task and when you're not so good at the task. So for instance, here, about 400 milliseconds or so into the cued target interval, you're about 5% better to make detections than 120 seconds later when you're at the trough, and then you go back to a peak and so on. So there is a systematic rhythm here. There's a fluctuation that's systematic, that's not random. We call it a behavioral rhythm. And that behavioral rhythm goes at 4 to 6 hertz, so it has some individual variability. Now, we trained monkeys on this task as well and looked at whether they have these behavioral rhythms or not. And to my surprise, they do. So this is a characteristic of attention function that has been overlooked for as long as we study attention, because we rarely ever, as cognitive neuroscientists, look at the dimension of time. We typically average over time and throw out that variable. But if you look at the dimension of time and how cognitive function unfolds over time, you see that it is dynamic. And I think this will turn out to be true for many, many other cognitive functions as well. The fact that monkeys who go back more than 20 million years in evolution with our species also have these rhythms tells you a lot about that they must have some kind of fundamental significance to attention function. And so that is what we are studying at the moment and try to understand. Now, I want to tell you a little bit about the neural basis of this. And again, I will show you recordings from the human brain. So here is our task, the schematic of it, and the recording that I'm going to show you is from a cognitive module in parietal cortex. So again, you can think about this as multi-unit activity, so activity from many, many, many 100 neurons or so that evoke some kind of activity here during this task. So let's go through this. Here we see the cue-evoked response. It's just a single trial, so these recordings in humans are extremely powerful. So a cue-evoked response. At this line here, this cue target interval, so this interval when attention is just allocated here, it's just a cognitive state, a cognitive operation, that's all what we're measuring here, starts here. And at this point in time, our target at threshold is presented. And then the green line here denotes the manual response, a button push of the patient. So in this case, the patient detected the target. And you can see right away that during this cue target interval, the activity is not flat, but it fluctuates systematically here at some kind of frequency that I will reveal in a moment. But I want to show you first that this is not just kind of one trial that we found this way, but that we can see that in all trials. So this is from the same recording and the same patient, and here just 2 other trials of different cue target, where the cue target interval has different lengths. So here, for instance, it's very short, and the patient is missing. So it doesn't matter, whether the patient is missing or detecting it. You always see these fluctuations. So let's take a look at the frequency at which this neural rhythm here that modulates this multi-unit activity is going. And it is a 4 Hertz rhythm. In the EEG literature, we refer to this as a theta rhythm. Well, now you have a behavioral rhythm that goes at 4 Hertz, and you have a neural rhythm at 4 Hertz. But that doesn't necessarily mean that they have anything really to do with one another. This could just be a coincidence. There are lots of theta rhythms in the primate brain. Now, we wanted to see if we could find anything in this neural signal, in this theta rhythm, that would make a direct prediction of behavioral outcome. What we looked at was how the phase of that rhythm relates to the occurrence of the target. So I want to go back to make this kind of intuitively clear what we're measuring here. So here the target co-occurs very close to a peak of that theta phase. This is also true in this example, but in this example where the patient is actually missing the target, co-occurs with a trough of that cycle that modulates this neural activity. And you can look at that systematically, and what you get is a curve like that. So this is the hit rate here, the change in hit rate, as a function of the neural theta phase, so that the phase of this rhythm here. You can see very clearly that there is a phase where, in this particular case, you get a large effect actually of about 6%. You're 6% better at this point in time, at this phase of theta, than at the opposite one. Now, we call these phase detection relationships, and this is a direct, the theta phase that we're using here can make a direct prediction about the behavioral success. So we link now behavior and a neural signal directly together, which is, always something that a cognitive neuroscientist is really excited about. So these phase detection relationships we find all across the network. So I show you here regions in the monkey brain, parietal cognitive modules in parietal and in frontal cortex, and also in that thalamic nucleus that I introduced to you, the pulvinar. And we see them as well, of course, in the human brain, as we just discussed, and here we can also look at a topography. So everything that has these very bright colors here has very strong phase detection relationships, like let's say something like this one here, and this is why it's bright. If it is more shallow, then it would fall into this middle range here, or if it's an antiphase, it would be in the blue range. And so you can see again and appreciate that the topography here, we're looking on the top, from the top onto this brain, is mainly in these cognitive modules in parietal and in frontal cortex. Now, in the human experiment, we saw another really interesting thing that took us a while to really kind of wrap our mind around. So here again, these phase detection relationships. This is the largest that we see in this patient. This is a little bit more shallow somewhere in between, but then we see also these regions that are in antiphase. And so we thought, okay, what if we just use the best phase detection relationship in this patient, that's this one, and correlate all the other recording sites and see what kind of correlations they, or phase detection relationships they set up and how they correlate with that largest one. And what we found was really, really surprising. So this analysis was able to parse this patient's brain into 2 different networks. One network, that's this frontal parietal network, is our attention network. Those are the cognitive modules that I've talked a lot about in this talk. And then in between, you get some other sensory and motor cortex. So you get the entire motor system that is an antiphase. So it's also predictive of behavior, but of poor performance. So the frontal parietal attention network and this motor, somato-motor network, they are in antiphase. And so what we think what's going on with these behavioral and neural rhythms here that we can study during attention function is that this is an alternation of 2 different states that the brain gets into, or this large-scale network gets into. One state is a more sensory emphasized state. That's a state when we can make more detections in the environment, and that is carried, by these regions in frontoparietal cortex, also in the visual system. And this is alternated with a state that is more motor emphasized, when we could move our eyes, for instance, to a new location in space. We could do a manual action, or we could do a covert shift of attention, which is a motor action for the attention system. Now, when we proposed this model of how attention works, people said that's too complex. A system cannot have these kinds of dynamics. There's way too complex to sustain. We think actually that these dynamics are the key to understanding the cognitive flexibility of this function and of this network. It can produce flexible behavior because it does not lock itself into one state, but it can set up these dynamic states, but just on the fly. So if somebody enters the room from behind the stage, you can direct attention immediately to that new person that will appear, because you have that opportunity every 250 milliseconds due to these alternating attention states that are going on. So we think that these kinds of dynamics that we now see also in other cognitive functions in cognitive neuroscience will really tell us a whole lot about what the neurobasis of cognition is. So we are really just at scratching very much the surface of, I think a very, very exciting development in this field. So let me summarize this part of my talk by saying that attention function unfolds over time and has rhythmic properties. Theta rhythms organize subnetworks, for instance, sensory and motor networks, into alternating states. Theta phase predicts both perceptual and motor performance. And this is a really important point to be made, as we also think that these rhythms here can set up a scaffold for other cognitive functions. So these dynamics could basically be used to also coordinate with other cognitive networks, like a memory system, for instance, and so on. So this could be a mechanism that serves a broader purpose that goes way beyond just supporting attention function. And that is the point of this last item here. Theta rhythms may serve as a mechanism to coordinate and integrate large-scale networks across different cognitive domains as well. So a few broader points that are highlighted today. Attention function is supported by a functionally specialized large-scale network. Attentional control is distributed and includes thalamic centers. And we think that the rhythmic properties account for the flexibility of attention function. Now, before I take questions and close my talk, I would like to alert you to this really important public outreach grassroots movement. Hundreds of scientists around the world are part of it. And I want to see if I can entice one or two of you today to join this too. Alfredo already talked about it, Frontiers for Young Minds. So this is a project that was founded about 10 years ago by Bob Knight and myself. We just started with the neuroscience section at that point. Now we have 8 different sections on life science topics. We started in 2013. It was launched at the Society for Neuroscience meeting in November. And at the end of the year, we had 19 clicks and views on the webpage. So we got a little scared. But we have now about 1.5 million clicks per year. So this has reached kids in all countries around the globe. The journal has been translated into major world languages. And we really think that this is a wonderful way to share our science with the rest of people in a way that's publicly accessible. The fun thing is that you, as scientists, write in a kid-appropriate way for children, 8 to 15, and the kids review you and give you feedback. And that can be very, very difficult. So yeah, if you are interested, please reach out to me, and I'll be happy to kind of give you pointers on how you can contribute. And with that, I would like to introduce you to my band. These are the many, many wonderful people, colleagues, with whom I had the privilege to collaborate throughout my career. I think I have learned so much from these collaborations, and this is, to me, the essence of science. And all of you will just be team scientists in this century, and this is where the fun really starts. So welcome to my band, and thank you very much for your long attention span.
Okay, we have a few minutes for a few questions. So if anyone has questions, you can walk to one of the two microphones.
Thank you, Sabine, for the wonderful talk. I have a question for you. So can this network be entrained and trained to work better?
Yes.
How?
Yeah, no, it's a great question. So those are studies that we are working on. We have done an experiment where we have and trained the cortical network from the thalamus, and at 4 hertz, so at that frequency that seems to set up these dynamic properties in the attention system. And to my surprise, we did not interfere with that cortical activity, but we made the cortical network more efficient. So we see faster reaction times when we entrain at a 4 hertz frequency from the pulvinar. And we also see that the detection thresholds get lowered. So we get better perceptual detectability as well. So there's a really interesting neural substrate to that. So the network dynamics get altered. So these are very brief pulses, so we do not get big artifacts and can look at what the cortical network is doing. And one characteristic is that the alpha activity that is very, very prominent in parietal cortex, in this parietal cognitive module, basically diminishes and is replaced by a theta rhythm that is very prominent typically in frontal cortex. And so it's possible that, you know, this increased theta rhythm and across the network mediates this kind of behavior. But that's very, very early times for that. So we just did these experiments a few months ago, but they could turn out to be really, really interesting. It's a great question. Thank you.
Thank you for your talk again. I wanted to ask a translational question about how attention and disorders in attention are often treated with neuromodulators that target, you know, the dopamine or norepinephrine neuromodulatory systems. Do you have any insights about how neuromodulators such as those may affect large-scale network communication dynamics or the presence or diminishing of certain frequency bands?
Yeah, we have very little insight. So we have done a little bit of work on that. Just at the behavioral level, we wanted to see whether methylphenidate would change these rhythmic structures that we see. And there's very little evidence for that. But I have not worked extensively with neuromodulation. However, what we try to understand is in the first place how the attention network develops. So I have projects on the neurodevelopment and also how this is different in kids with ADHD. So I'm very passionate about kids, you know, who struggle with attention and other cognitive abilities, and we really don't understand that. We just pour these drugs on these developing brains with little understanding of how this development looks in the first place. And so we are studying that at the moment, but I cannot tell you much yet about the results. But that is, again, you know, an interest we have, because coming back to that entrainment, if it would turn out that some of these kids, and I think it will turn out that way, actually are characterized by not having these rhythmic dynamics that I've shown in the last chapter of my talk, but they basically lock into one or the other state. You can think about that motor emphasized state as a hyperactive state and the sensory emphasized state as a hyperfocused state. Then basically reconstructing or reconstituting these rhythmic structures and dynamics could be a real therapy that is driven and based on brain science, and that would lead to something like an entrainment therapy, but not the drugs that the kids get at the moment. So yeah, that's a very big topic for me. Thank you for asking. And eventually we will get there and really do the proper neuromodulation as well, so that we can understand that.
Thank you very much for a wonderful talk, and your data, especially towards the end on the brain dynamics, are very, very exciting indeed. I'm wondering whether affective states could be playing a further modulatory role on the frequency. So whether in a depressed state you might see a slowing down of that 4 to 6 theta band frequency that's modulating the entire network, or in a state of acute fear, you might see an increase in that frequency, so that there might actually be additional intra-subject modulatory effects that could be guided by affective states. And then that makes me wonder whether interventions that are based on transcranial magnetic stimulation or something, to the extent that they are effective, are tapping into restoring those kinds of dynamics that you discovered. Thank you.
Yeah, I think almost for sure for TMS that is true, that they are tapping into these kind of rhythmic structures that probably all cognitive networks have. And yes, I mean, there are probably a lot of factors that can modulate the frequencies of these rhythms. That is also something that we're actively studying and trying to understand what the extreme conditions are, if you like. We have a model of these dynamics together with Nancy Coppell. So Nancy, who is a computational neuroscientist, has modeled this carefully. And in the model, we cannot go slower than 2 hertz and not faster than 8 hertz. And then the model will not produce any meaningful behavior anymore. So we do not know whether that will, turn out to be true for primate brains as well. But that gives us at least, some hypotheses to work with. But that's also something that we're very interested in. Yeah.
Okay, so please join me all to thank once again Dr. Sabine Kastner for the wonderful talk.